Well, hello everyone. I'm Michel, and it's a real pleasure to have you with us today. So thank you for joining us today and taking the time to spend with us for this session. In this webinar, we'll be hearing from Dr. Briana de Miranda, who will be talking to us about how the environment affects Parkinson's disease, and more importantly, what you can do about it. Dr. de Miranda is a neurotoxicologist and associate professor of neurology at the University of Alabama at Birmingham's Killian Center for Neurodegeneration. Her work focuses on how environmental exposures contribute to neurodegenerative diseases, integrating toxicology, pharmacology, and neuroscience to develop strategies for prevention and treatment. So as always, before we begin, and you know that, please note that this session is for informational and educational purposes only. So if you're seeking medical advice, please consult a qualified healthcare professional. As always, again, there will be time for questions at the end of the presentation. If you would like to ask a question, please use the Q&A function located at the bottom of your screen. Please do not use the chat function, use the Q&A, please, for your questions. So for those joining us for the first time, No Silver Bullet is co-managed by Mark Lambert and myself, and our goal is to share Parkinson's expertise and help our community make informed choices about their health. We share all our webinars on YouTube and on Spotify, and you will find the links in the chat and on our website. Our website is nosilverbullet4pd.com, nosilverbullet4pd.com. But let's now get started. Please join me in welcoming Dr. de Miranda. Briana, the stage is yours.
Thank you so much. Well, it's a pleasure to be here, and I'm really thrilled to be able to talk about this topic today. This is the main research topic that I conduct in my research lab here at UAB. And I'm really excited to be able to give you a little bit about my perspective. As Michel mentioned, I am a toxicologist, so I'm not a neurologist, I'm not a clinician, or even a biologist. I have formal training in toxicology, but I've always studied Parkinson's disease. And so I hope what I can do today is give you some information about how myself as a toxicologist and other toxicologists would approach this disease. And I'm going to talk a little bit about how this disease is going to be, I think, changing a little bit in how we study it from today moving forward. So please join me in this. Parkinson's disease, as many of you have heard, is a disease considered of gene-environment interaction, and that's not necessarily unique among diseases, right? So Parkinson's falls along a spectrum like this, where environment can contribute to certain diseases and genes contribute to certain diseases, but most diseases are a combination of genes and environment. However, this is not only true for Parkinson's. This could be true for cancer or heart disease or diabetes, but especially for Parkinson's disease, because this is really critical in its relationship to how etiology matches presentation. So for example, at one end of the spectrum, we have individuals who may have monogenic or inherited or familial Parkinson's disease. But at the other end of the spectrum, we also have individuals who have worked with chemicals that have directly led to the development of disease. And so you can see that somewhere in the middle here, Parkinson's incidence along this incidence curve is really a combination of both those genes and environment. So most people don't have a really clear genetic link, and most people don't have a clear environmental link, but it's somewhere in the middle. So what we call Parkinson's incidence or idiopathic Parkinson's disease really refers to gene-environment interaction. And you can see that it's pretty complicated. So things like polygenic risk as well as epigenetics also play a role into this. But just the key take home here is that idiopathic Parkinson's disease is really a gene-environment interaction. So environmental risk factors play a role in all Parkinson's disease, just as I mentioned. But another way we can look at this is through heritability. So Parkinson's disease is a mostly idiopathic disorder. About 36% of Parkinson's can be explained in some fashion by heritable factors. And this includes about 10 to 15% of monogenic or familial Parkinson's disease. And what this really means is that in about a third of cases, there is at least some genetic risk factor present, but it doesn't mean that that was necessarily the cause of all disease. Parkinson's, as you probably know, is the fastest growing neurologic disorder, and it is outpacing the rate of aging. And so we believe that at least some of this outpacing of aging is due to environmental or industrial byproducts that are toxic pollutants in the environment that are increasing risk for Parkinson's. Another way we can look at this is by using a metaphor. So in this example, this is an ocean where the ocean floor is your intrinsic or genetic risk, and everything above that in the water is your exogenous risk or environmental risk. And so you can see that some people might start with more or less genetic risk, but it usually takes at least some amount of environmental or exogenous risk to reach that threshold for Parkinson's disease. And then once a person has Parkinson's disease, they're going to continue to progress. And phenotype, the way a Parkinson's presentation happens in the clinic or as it happens to us in individuals, this is going to be also affected by both gene and environment interaction, as well as the way that Parkinson's progresses. And so the key take home here is that both genes and environment can also contribute to Parkinson's progression and phenotype. So I think something that's really unique about Parkinson's disease is that we've known that toxic exposures play a key role in Parkinson's disease, and we've known this for many years, so over 100 years. The very first environmental link to Parkinson's was the H1N1 influenza pandemic that happened in 1918, 1919, where people who had viral encephalopathy developed Parkinson's disease in the years following. In the 1950s, we knew that manganism, a type of movement disorder that happens when you are exposed to the heavy metal manganese, was predominantly found in welders. And so this is something that occurred in sort of a cluster. People who were using welding rods with manganese in them developed manganism. Now, we've mostly taken manganese out of welding rods, and there are now better methods and better PPE for welding, but this is a problem that did happen up until probably the late 50s, early 60s. We started finding Parkinson's clusters in the 1980s, and this is really unique because most diseases that are thought to be idiopathic do not cluster. And when we see clusters, we typically look for an environmental source. So for example, people who injected themselves accidentally with the neurotoxin MPTP were found to be extremely Parkinsonian very quickly. I'll talk again about that in just a moment. And we also found in the 1980s that these clusters were unique, but not necessarily unique in epidemiology. So our large-scale epidemiological studies found that rural populations in some Western nations and urban and industrialized areas in China were associated with increased risk for Parkinson's, again, in thought because it was related to environmental exposure. So talking a little bit more about MPTP now. MPTP is a synthetic chemical. It is a derivative of heroin, or it's a synthetic type of heroin, and it was accidentally injected by some individuals in Northern California in the 1980s. You may have heard about this story before. MPTP really led the way for us to understand scientifically some of the mechanisms underlying Parkinson's disease. So for example, MPTP is a very specific type of mitochondrial toxicant called mitochondrial complex 1 inhibitor. And it led the way in the 1990s for us to sort of uncover how complex 1 is involved in dopaminergic neuron degeneration. In fact, we found that MPTP is specific for dopamine neurons, and it specifically blocks complex 1, which is really critical for dopamine neuron function. In the 1990s, we started to get a better idea of gene-environment interactions. And as we move forward into more current times, we found that it wasn't just pesticides or synthetic chemicals, but actually solvents and head trauma, as well as potentially air pollution and different mixtures that were epidemiologically linked to Parkinson's risk. Now, as we get into the current state, we're now using more advanced tools to try to understand how Parkinson's disease is related to environmental exposures, which exposures are most critical for the disease, as well as the complex interactions between different chemicals, sometimes referred to as the neural exposome, as well as our own body's response to them. So I'll just make a quick note as I end our timeline here in 2025 that we are still really in our infancy of understanding the true role of environment and Parkinson's disease. And so even though we've known for over 100 years that this has been happening, we still have a ways to go, and it's something that's really critical to keep studying. So what are the key environmental risks for Parkinson's? Pesticides, solvents, head trauma, air pollution, and heavy metals really make up the majority. Pathogens, certain viruses have been linked to Parkinson's disease. And then emerging contaminants like microplastics or PFAS chemicals have been implemented sort of insinuated to be risks for Parkinson's. However, we don't know for sure that these are going to emerge as genuine risk factors. And so one of the things that like my lab's trying to do is understand how things like pesticides and solvents work to cause dopaminergic neurodegeneration, to cause those Parkinson's hallmark pathologies. Therefore, we can apply that to things like emerging contaminants and really start to understand what is going to be a true risk for Parkinson's disease. So I'll switch gears a little bit now and talk about how these risk factors work to not just induce risk for Parkinson's, but also heterogeneity. So as you might imagine, what you're exposed to matters. And the reason for this is because different exposures have different mechanisms, right? So if you take the chemical paraquat, this is a broad spectrum herbicide, and compare that to the organic solvent trichloroethylene, you can see here they look functionally very different. And they have different mechanisms, different toxic mechanisms. However, they are both risk factors for Parkinson's disease. So understanding how they are both similar and different can help us understand how Parkinson's pathology develops from these exposures. But each chemical has different eliminations and half-lives, and their potencies can differ. And so what you're exposed to can matter. How you're exposed can also matter. So most chemicals or environmental risk factors for Parkinson's can be exposed via inhalation, ingestion, or dermal exposure. And so we think a lot of Parkinson's risk factors are either inhaled or ingested. And in fact, there are some hypotheses that if a toxicant is predominantly ingested, that it may kind of insinuate a body first or initiate a body first phenotype starting in the gut. And if it's predominantly inhaled, it may be more of a brain first or an olfactory bulb initiating disease. So this is just a hypothesis, but an example of how the environment can affect more than just Parkinson's risk. It can actually affect the phenotype someone may present with. There's often also a notion, I think, for some people with Parkinson's that an exposure might have happened when they were younger and then they developed the disease later in life. And this is definitely something that I think has a lot of validity. There's just not a lot of data right now for us to understand this, especially in the human population. There's a lot of experimental data on this, but it's important to note that exposures can happen all across your lifespan. So it can happen early life, it can happen all the way through your working adulthood, an occupational exposure, and even into an aged adulthood. But you are more vulnerable in terms of neurotoxicity during neurodevelopmental periods as well as aging. And so there are some examples of this. This is really early data to suggest that early life exposures to pesticides or living on a farm and drinking well water is associated with Parkinson's disease risk later in life, as opposed to living on a farm or drinking well water as an adult. And so I think this makes sense for a lot of people because Parkinson's disease generally appears later in life. And so you've had, a long time of your lifespan to develop the pathology of the disease. And then lastly, just to make a brief comment on gene-environment interaction, even though this is a really complex topic, there are several different types of genes that can be risk factors for gene-environment interaction that relate to Parkinson's disease. For example, the enzymes that metabolize xenobiotics or metabolize toxicants as they come into our body. Risk genes for Parkinson's, like the LRRK2 mutation, for example. Enzymes that detoxify chemicals, as well as blood-brain barrier transporters. So just a brief note about each one of these. For example, the cytochrome P450 enzyme CYP2D6 is associated with different variants are associated with risk for Parkinson's disease. This is a xenobiotic metabolizing enzyme. My own work has looked at the way LRRK2 can interact with different environmental contaminants, including something like the solvent trichloroethylene, as well as rotenone, and kind of cause risk for Parkinson's that way. Glutathione, which is a detoxifying enzyme, is associated with different polymorphisms have been potentially associated with Parkinson's disease risk. This makes sense because of the way that it detoxifies different toxicants. And then the blood-brain barrier transporters, like the ABCB1 gene variant, has been potentially associated with Parkinson's risk. And this is because these are the channels that actually export toxicants across the blood-brain barrier. And so these would be important in getting different toxicants out of the brain. And you could imagine if you have different functionality in that blood-brain barrier transporter, it could retain toxicants in the brain for longer, therefore initiating more toxicity. So then what is it that makes a chemical a Parkinson's toxicant? So if you're exposed to thousands of chemicals a day, what are the things specifically that we worry about for risk for Parkinson's toxicity? In order to be classified as a Parkinson's toxicant, a chemical or an environmental risk factor has to be able to cause the selective loss of dopaminergic neurons, either by accumulation or specific toxicity at the dopaminergic neuron. So for example, MPTP binds to the dopamine transporter, or the toxicant has to cause some kind of pathology that dopamine neurons are selectively vulnerable to. For example, mitochondrial complex 1 inhibition. So say a chemical enters the body and it sort of broadly inhibits the mitochondria. Dopaminergic neurons are selectively vulnerable to mitochondrial inhibition, and therefore they may be vulnerable to death because of that exposure. Other ways that environmental toxicants could lead to this type of pathology is neuroinflammation, as well as alpha-synuclein aggregation. Again, a toxicant causing misfolding or aggregation of alpha-synuclein. We also see that certain toxicants could be associated with degeneration of extranigural areas, as well as motor and non-motor symptoms. Now, molecularly, I've already talked a little bit about mitochondrial dysfunction, so I won't go into too much more detail on that, just to note that mitochondrial dysfunction, as well as the emission of reactive oxygen species from the mitochondria, is thought to be a key role of environmental toxicants associated with Parkinson's. There's a lot of research going on about this right now, and so how this works over a lifespan is currently sort of under investigation. But another one I would draw your attention to is the interaction with alpha-synuclein. And so are there environmental risk factors out in the environment that we're exposed to that cause the acceleration or exacerbation of ASIN misfolding? I think this is an area of study that's really critical right now. This is something that my lab studies, is this dysfunction of endolysosomal pathways, so how alpha-synuclein is broken down in the lysosome. And so all of these things together, either in one part or another or combined, can ultimately be what causes neurotoxicity from an environmental exposure that is related to the specific pathology of Parkinson's disease. So let me take a moment and talk to you about some examples of this. I'm going to use an example first from my lab with the organic solvent trichloroethylene, or TCE. TCE is an organic solvent. It is used in industry. It's predominantly a degreasing agent, but it's used in other ways as well. And you get exposed to TCE in a number of different ways. The predominant way is via inhalation. So if you work with TCE, you may inhale it as you're using it. You can also be exposed to TCE if it gets into the environment and pollutes or contaminates the soil or groundwater. It can get into groundwater aquifers and become a vapor intrusion hazard into residential and commercial buildings. It can also be a drinking water contaminant. And so you can imagine that if it were in your soil and it were off-gassing into your house, you may not even know that you're being exposed to this. In fact, most people do not. If they have TCE contamination, it's usually found potentially years after the contamination started. And so it's really a strong risk factor in heavily polluted areas. In the United States, which is where I gather most of my data, the map that you're seeing at the top of your screen is from the Toxics Release Inventory. This is sort of just a crude depiction of where TCE was reported to be released. And this is really underestimating TCE release, but hopefully what you can see is that over time, we have reduced our use of TCE in the US. This is true around the world as well. And this is good because TCE, is a toxic chemical that's implicated in Parkinson's risk as well as in cancer. But unfortunately, with TCE, it is a chlorinated compound, and so it does not break down very well. And so a lot of the TCE that we emitted even 50 plus years ago is still in the environment. I will note that as we ramp down use of TCE in the United States and in Europe, there are some countries that are still using TCE. In fact, their use is growing. So for example, Central and South America, as well as Southeast Asia, are really using TCE in heavy quantities right now. One of the more famous examples of TCE environmental contamination is at the Camp Lejeune, North Carolina Marine Corps base. This is a base that was contaminated through a dry cleaning site, and individuals who lived on this base were exposed to TCE as well as other organic solvents used in dry cleaning. And individuals who were exposed at Camp Lejeune have a 70% increased risk for Parkinson's disease. We now know that ambient TCE exposure is also associated with increased risk for Parkinson's. There was a paper that was just published last week showing that individuals who live near factories that emit TCE, for example, this factory in Oregon, have increased risk for Parkinson's in sort of a radial and wind-related direction from TCE exposure. And so we're getting better data on this. We're still not quite there in terms of looking at global risk for Parkinson's from TCE exposure, but I think it's something that we're really honing in on, and hopefully we'll get a better idea soon of just how much Parkinson's risk is really coming from this.
Miranda, may I please, so Briana, may I interrupt you for a split second? If you go back to the previous slide, Briana,
Yes.
Do you mind just telling us how to understand the 1.68? I mean, some of us will be able to understand that. I don't actually.
Yes, so that's a really good question, and thank you for letting me do that. Let me quickly reshare my screen here so that I can just get my view back that I want. So the odds ratio of 1.68 refers to a calculated ratio above 1. So 1, if you have an odds ratio of 1, then there is no additional risk for disease beyond just chance, right? The odds ratio of 1.68 indicates that you are about 0.7 times over that 1. So that translates to 70%. So if 100% risk were the total, you have about 70% increased risk with an odds ratio of 1.6.
That's very clear. Thank you very much.
Great. Yeah, thank you for asking that. And I think that's a really important thing to note, because when you talk about odds ratios in just single digits, it doesn't maybe seem so impactful, but when we think about it in percentage, it is really a clear thing. Okay, thank you. So now moving on a little bit to what I actually do in my lab. So this is where my lab focuses. We've developed models of TCE exposure in lab animals, so rodents, using rats and mice. And what I'm showing you here is an example of TCE exposure with ingestion. So I mentioned ingestion and inhalation are the two routes that we predominantly think of for exposure for Parkinson's disease. TCE is predominantly inhaled in the environment, but ingestion is a concern as well. So as you see, with 6 weeks of a relatively high dose, 200 milligrams per kilogram of TCE, in rats, my lab was able to recapitulate the loss of dopaminergic neurons in the substantia nigra. And you can see this here in red. So hopefully you can see my pointer here. This is the substantia nigra in a healthy rat. In red, you can see the dopamine neurons in the SN and their terminal projections into the striatum. But in the TCE exposed animals, you can see we have a loss of dopaminergic neurons here in the substantia nigra, and then a pullback of the terminals in the striatum. We can also see this with inhalation, but I want to note that we used a much, much lower exposure level for this. So only 50 parts per million of TCE exposure in rats, which is actually 1/2 of what the United States says is safe to be exposed to at the workplace. So if you work directly with TCE, there's a 100 parts per million exposure limit per day to TCE. So at 50 parts per million in rats, again, hopefully what you can appreciate is we see the loss of dopamine neurons in the substantia nigra and their terminal projections in the striatum. And so this is recapitulating that hallmark pathology of dopamine neuron loss that I talked about a moment ago. Another thing that's important to note is TCE is not specific for dopamine neurons. It does not accumulate in these cells in any meaningful way. It's simply exerting pathology in the brain that dopamine neurons are susceptible to. Another example of this is the organic pesticide rotenone. So rotenone is found in nature in the stems of jicama plants and some other legumes. And it's a very large structure in comparison to TCE. And at one point it was used, I think throughout the world, definitely in the United States, to garden or to kill off invasive pests inside of agriculture. However, it's been banned by the USDA. It is still used widely to kill off invasive fish species in lakes and streams. And so that's what this manual in the middle is showing you, how to properly use rotenone. And you can see some people are using proper PPE here while they're applying it, but there's some concerns, I think, of using rotenone in the water and how that might be an issue, particularly for people who apply it. Now, rotenone is associated with increased risk for Parkinson's. This is an epidemiological study at the right conducted by Dr. Caroline Tanner, and you can see that they found an odds ratio of approximately 2.5 for rotenone, so over 2 times higher, 200% higher risk for Parkinson's with rotenone. In the lab, we can actually recapitulate this again, like I just showed you for TCE. If you dose rats systemically with rotenone, you can see similar pathology, which is the loss of dopamine terminals in the striatum, as well as loss of dopamine neurons in the substantia nigra. And so the reason I picked these two to show you is that there's a clear epidemiological link between TCE, rotenone, and Parkinson's disease, and we were able to recapitulate this with experimental models in the lab. And that's really important because in order to show that something is really a bona fide risk factor for Parkinson's, you have to gather data in multiple different ways. So is it linked epidemiologically? Can we recapitulate this in the lab? And then what are its mechanisms? And so that's really critical for understanding risk factors for Parkinson's. So as a summary from this first part, Parkinson's disease risk is very complex. It includes both endogenous and exogenous risk factors. Age is also really critical, as I noted, for exposures across the lifespan. And then what I didn't talk about, but something that should be noted, is that there's a degree of chance here too. So every day we have different exposures and different things. There are proteins folding and misfolding in our bodies at any given moment. And so, just stochastically, we may find that pathology arises kind of as it builds upon these other risk factors. So something to note there. I have this map of incidence in Parkinson's disease in North America, and I sort of apologize for making this a US-centric presentation. This is where I gather data from and what I have most access to. But in some ways, this is where we have the best data for really granular data for clustering effects. And so on the one hand, what's really interesting to note about this is that Parkinson's is not uniform across the US. You would expect it to be if it were purely a disease of genetics or chance. Maybe not so much genetics if there's familial clustering. But I think what's probably apparent is that these are areas of heavy industry. The Central Valley of California, the Appalachian Mountains, and the industry in the Great Lakes region, the oil and gas region in the Gulf, and then the industry across the eastern seaboard. The second point of this would be we need data like this from around the world so that we can really just start to understand what risk factors differ from different countries, right? So where pesticides could be a big problem in the US, there might be another risk factor somewhere else. So what can we do to mitigate these exposures? This is something that we talked about when I was talking with Michelle and Mark about coming on today. They asked if there were some practical approaches that we could take to reduce our exposures. And so I'll try to go through some of those now. I do want to give a disclaimer that I'm not an expert in these engineering systems. I'm actually on the opposite end of this, right? I expose animals to these chemicals to try to understand what's happening in the brain. But I do, just like all of you, I am also a consumer, and I want to understand the way that these work and the way we might mitigate our risks from environmental exposures. I will note I'm going to be very careful not to give any specific product recommendations here. So I will encourage you to look up these on your own. And if you have any questions, we can go over this at the end. So what can we modify in our environment? This is really just a few things, right? So water, air, diet, and then the rest is sort of specific to what an individual might be doing. So do you spray something on your garden that's a pesticide? And then lastly, can we do better with our policies or our information gathering in order to modify things at a more population level? So just going through this individually. Water is probably the most well-known in terms of filtration. So I look for resources online to try to help understand, number one, what is in my water and what do I need to be aware of when I'm filtering it out? So I used the Environmental Working Group's website to search Birmingham, Alabama, where I'm located. And I found that my water source, the Birmingham Water Works Board here at my office, contains 27 contaminants that were detected and 11 that exceed the Environmental Working Group's health guidelines. Now, this is the Environmental Working Group's guidelines. EPA might have a different guideline, but it gives me some recommendations for how I might want to filter my water. So for example, it says if I use a carbon filter, it will get 9 of those contaminants. But if I use a reverse osmosis filter, it will get 11 of these contaminants. And so pros and cons of this, of course, cost as well as some of the specifics of running these systems. So just to give you an example of what these look like, there are different types of sediment and carbon filters. Sometimes granular activated carbon, like a Brita filter or something in your refrigerator, are the most common. I think a lot of people are familiar with these. A lot of you might also be familiar with reverse osmosis. So this is another way we filter our water. But what you might not be familiar with is that you can actually do reverse osmosis in your entire house. So if you're an individual who lives in an area with really, really high contamination and you know that that contamination is a concern, this is something that people would be interested in, is really purifying the water in all of your home. And why would you want this? Because you don't just drink water, obviously, we also cook with it and bathe in it. And so if water contamination is a serious issue, there are solutions that can get at whole home systems. Now, of course, this is going to be specific for a person who already knows they have a lot of contamination. No one would probably want to go about this in this way if you were just concerned about your water. So getting your water tested if you have concerns, or making sure you know where your water, what's in your water at your location, is important. Air quality is another relatively simple thing to measure. This is going to be predominantly particulate matter. So air quality indices throughout the world measure different things, but mostly it's going to be the most common contaminant, and that's usually PM2.5 or PM10. You can see that European Air Quality Index, this is an online website that you can go to look at the air quality index all around Europe. And you can see most of it's green and yellow with some spots of red. There are also apps you can put on your phone. So for example, this is an app called Pacu, which is related to the monitors that are called Purple Air. This is sort of unique in that it takes monitors that people, or consumers rather, have in their homes or businesses. And so each one of these hotspots that's located here actually comes from consumer data. So this is the collective air monitoring from people who have these Purple Air air quality monitors. You can also use things like EPA. If you're in the United States, this is a free app, or you can use their website that measures air quality indices throughout the country. So for example, in Los Angeles on October 3rd, when I last pulled this data, the air quality index was 52. So how do you interpret this? That's going to depend on where you are and which air pollutant is most concerning. But the general, I think, suggestion is that when things get into the orange and red, you want to avoid time outside. You would definitely want to probably avoid exercising outside, because this is when you're going to have a lot of issues with cardiovascular concerns from air pollution. And you also may want to close your windows and make sure you're running air purifiers during those times. So a lot of different things for air quality, and there's also different ways to purify your air. So for example, most air purifiers contain a HEPA filter as well as a carbon filter, and they have different and specific purposes. A HEPA filter is going to get out things like bacteria and viruses, pollen, and PM2.5, whereas carbon filters might get out VOCs, particularly odors and things that cannot be trapped by the HEPA filter. And so a good air filter would have both, and there are so many air filters out there, it's very difficult to choose any, but I'll just talk briefly about the different types. There are mechanical as well as electronic air cleaners. Most people are going to be familiar with the mechanical air filters. This is the type that I use in my home and office. But I highly recommend looking at the EPA's Guide to Air Cleaners in the Home. It has every piece of information you probably want on this. I actually found it super interesting to go through. There are so many different types of air filters. Again, most are going to be mechanical, but there are air cleaners that actually use things like UV light and different ozone generators in order to, as they say, clean the air. But there's an important thing to note about this, which is that the oxidation-based air cleaners, those chemical cleaners, can actually be really, really highly variable. And so There are different groups who have tested these. This is a published paper from 2021 showing different VOC emissions and how they are removed by these oxidation-based air cleaners. So what they did in this study is they took toluene and limonene, which are two VOCs. They put a known amount of these VOCs into the air, and then they tested air filters and saw how well they could clean these from the air. And as you see in the figure to the right, these different air purifiers had a lot of really different response to these VOCs. Some removed them from the air really well, some did not. And so important to note that not all air purifiers are created equally, and to do your research on which ones might be most important. But that being said, there is an actual and sort of reasonable reason to want clean air in your indoor air. And this is because the air can actually affect us in our daily lives in terms of our health in ways that, it's really hard to kind of guess for other chemicals. So PM 2.5, which was studied in this paper, shows that if you reduce PM 2.5 in a moderately polluted area with a proper air filter, it actually lowered systolic blood pressure in individuals with the air purifier versus the sham control. And so why is this important? Because blood pressure can affect many things. So we might think cardiovascular disease, but exposure to PM 2.5 or different air toxics are a concern throughout our lifespan. So not just for Parkinson's risk, but say you're a person with a chronic health condition and you would like to not exacerbate that health condition, you might want to take into consideration that what you're breathing in now, even if you already have a chronic condition, can actually sort of act to affect all of your health, right? And so this kind of gets at that idea of, is there anything we can do to stop progression? That's pretty difficult to say with an environmental exposure. We don't know if stopping environmental exposures would stop progression, but you can say, well, I'd like to at least keep myself as healthy as possible now. And one of the ways we know that can be true is by filtering our air. So thinking back about just kind of good habits, our diet is another source of contaminants. So one of the main things that we think about is pesticides on our fruits and vegetables. And washing fruits and vegetables is actually a really great way to get pesticides off of them. And so sometimes you probably have a habit at home. Maybe you're really good about washing your fruits and vegetables, but the National Pesticide Information Center has great guides on how to actually wash fruits and vegetables properly. And so this website's really great for that. There's actually been studies that look at how we wash our vegetables and how many pesticides can be removed. This is for leafy vegetables specifically. I found this really interesting. Actually, if you'll note in the table down here, running water was one of the most effective ways to remove pesticides from these different leafy greens. And so something to take note of, make sure that you're actually washing pesticides off in the most efficient way by knowing how to best wash your fruits and vegetables. There's also another dietary source of neurotoxicants, which is heavy metals. The risk here mostly comes from supplements as well as fish. You've probably heard that methylmercury is a risk factor in fish. I'll talk about that in a moment. But in terms of supplements, the heavy metals we worry most about are arsenic, cadmium, mercury, and lead. And this study that I am showing up here now is from actually protein powder. And so what this group did is they looked at different protein powders and they tried to assess how much excess heavy metal was in each of these protein powders. So from this paper, they found that arsenic, cadmium, lead, and mercury were found in some, but not all, of these protein powders. And they estimated how much daily intake of heavy metals that would be if you had 1 or 3 servings of this protein powder per day. An important note about this paper is that they did not find, at least in their results, that these heavy metal levels were a concern for health risks, but just something to be aware of. Heavy metals do exist in our supplements. There are other reports, particularly in Ayurvedic medicine and some other supplements that have been reported to be contaminated with heavy metals. So this is actually a contamination issue and not necessarily something that's naturally occurring. So always good to know if your supplements are being tested by a third party. I also mentioned fish. We've known for many years that methylmercury is a concern in fish. And as you'll note in this infographic from the US EPA website, there are some fish that are known to be really high in mercury. And so at the bottom right, you see that things like orange roughy and swordfish are high in mercury, and that there are other fishes that are potentially lower in mercury that could be good to swap out. And they have guidelines for serving so that you can know whether it's appropriate to eat these in small quantities or how you might want to gauge your fish consumption. This is always so difficult, right? And I'm not a nutritionist, but it's difficult because on the one hand, fish are healthy for us and they provide certain nutrients that are considered beneficial to our health. And so you want to always balance these things with, risks for exposure. And so, certainly do not stop eating fish, maybe eat salmon instead of swordfish if you must, but, always in moderation, I think is a key role. Okay, getting into more specifics now, if you are a person who gardens or who works with chemicals, always make sure you're wearing proper PPE when you do so. It might seem like a lot to be using safety goggles and a mask if you go out into your garden to spray chemicals, but it's actually probably the minimum we should be doing in order to make sure that we're being safe. Most people, what they may not know is that you should never bring clothing inside of your house if it's been used to spray like pesticides outside, so keep it separately. And then I found that one of the most easy to digest guides on pesticide spraying and respirator wearing is from the University of Maine. This is from their website on the right, and they talk about the types of air purifying masks that you're going to want to wear if you spray pesticides. So if you're in a garden, if you're an avid gardener and you're spraying pesticides around, you may want to invest in the right type of mask. And I would refer you to resources like this in order to understand that. And remember, as you probably know from our recent experience with the COVID pandemic, respirators have to fit properly in order to work. And that's really true for things like pesticides because they're a lot smaller than viruses and they can get through the filtration masks really easily. Okay, so kind of as we get down to the last bit here, indoor air monitors is really the last sort of personal thing that I have on this list. These are an emerging technology, and I will note again, kind of like the air purifiers, indoor air monitors are going to vary widely based on what they are and how they work. Again, the EPA has a very, very nice published website on this. So low-cost air pollution monitors and indoor air quality, I highly recommend looking through this guide if you're interested. They go through a lot of great data and they can give recommendations. I also want to note that there is no currently accepted air concentration limit for most of these indoor air pollutants. And so if you are searching for making sure that your indoor air quality is good, you may have to do some digging to understand what is considered safe and healthy versus not. So just a caveat there. However, we do know that different air purifiers or different air monitors have different functional capabilities. So this is a nice published paper showing the performance assessment of these low-cost environmental monitors. I'll just note, as this busy image on the right shows, different VOCs that were emitted specifically by the group testing this. And then these are the results of the different air monitors that picked up these things like candle burning and popcorn cooking, which I think is a really interesting way to do research. And I wish I could incorporate that into my lab more often. But you'll notice that on the left-hand side, you can see that some of these air monitors really pick up quite a lot of the VOCs that are emitted. And so TVOC, total VOC concentration, is something that is important to note if you're looking for an air monitor. TVOC, PM2.5, PM10, and CO2 are often things that are listed. Now, this paper did go through and actually rank some of these different air monitors. And so this gives us some idea of which air monitors are doing a relatively good job of measuring which environmental contaminants. Again, going to refer you to these online resources if you have any specific questions about what might be right for yourself. In the idea of information gathering and monitoring, we're really just beginning, I think, to understand how we can gather the large scale data of everything that we are exposed to in a day in a week, in a month, in a lifetime. And so commercial labs are starting biometric testing with, they're using reference levels by measuring things like blood and urine, and they'll tell you, you've exposed to this much pesticide, these different toxicants. But it's important to note that we don't have specifics for neurotoxicity yet for these commercial testing labs. And so you can get this done, but it may not be that informative to you overall in terms of neurotoxicity unless you're engaging with a specialist. There is a great initiative being undertaken using silicone wristbands for passive monitoring. One of my colleagues at Emory, Doug Walker, has an ongoing study using silicone wristbands to try to get at the entirety of what a person is exposed to over a certain period of time. This is, of course, very experimental still, but I think there are some commercial labs out there that will potentially be willing to sell you a wristband and monitor it for you. But of course, I would caution you that this is still mostly experimental. And then there are professional organizations that are designed to assess exposures. And this is a developing area, but I expect we'll see more as we integrate more of environmental risk into our total health. And so lastly, I'll just make a note about how we can integrate this all together in order to affect environmental risk for Parkinson's on a more population-wide scale. This infographic comes from a paper that I published with some colleagues in 2021. And we came up with what we felt like were really critical missing areas in both the basic research science area as well as clinical science. But what's most important and the key take home from this is that we have to keep researching the environment and Parkinson's disease in order to really make an impact. I presented to you today some examples and noted that we probably need to do more. The most important thing we can do is advocate for this research to be conducted, both the research that I do in my lab as well as epidemiological research. And in that sense, we can sometimes turn this into policies that actually protect us against these environmental risks associated with Parkinson's. And so that's something that I think we can do as a Parkinson's community. In fact, the National Act to End Parkinson's Disease in the United States was passed a few years ago. And some of the provisions in there, I think will, or at least they had the intention of trying to understand policy from environmental exposures and how we can mitigate risk. And so there are definitely different levels that we can undertake, but it's going to be really variable based on where you are and what's already implemented. So as a summary, I'll just remind you that all disease is a combination of genes and environment, not just Parkinson's, but also, every other disease that's out there, particularly chronic diseases. Your risk is never zero. So, don't take home any anxiety from this presentation. You want to make sure that you understand that we are all just trying to do our best to mitigate any disease risk. Some environmental exposures could influence progression. So if you already have Parkinson's disease, you may find that some of these are beneficial. Others you might say, I don't really care because I already have Parkinson's disease. But I think it's really important to note that we can improve our health and potentially progression if we're really careful and methodical about this. And then pragmatic approaches. I noted that a lot of the new technologies out there are still emerging. And so you may want to explore some of these, but you also are going to want to know that there's going to be some variation across the spectrum here. Okay, so thank you very much for letting me get through that. And I look forward to talking with you more now.
Briana, thank you very much to you for this presentation that I must say was very impressive. In particular, I would say in the bit that you were a tiny bit less comfortable, which is not part of your daily life, which is what can you do about it? And I think you did a brilliant job and gave us a full range of things we can do, not only to protect ourselves against the acceleration of our condition or the progression of our condition, but just to stay healthy as we all age. And I think that all your advice was extremely well thought through, and you very kindly also gave us the sources where we can go to get more information. So thank you very much for this. I see we have some questions there. Let's just have a look. So I'll go through those with you if you want. The first one that I see there, okay, we can just go up the whole list.
Okay.
Yes. So Leah is asking about your recommendation concerning drinking water. She has had a well, so she drank only from the bottled water the last 8 years. And today she bought a reverse osmosis machine. You mentioned that in your slides, to basically to drink her own town water in her new home. She wants to get away from microplastics. I know you covered that topic, but actually, I've looked myself at reverse osmosis machines, and I could see that they filter out also a lot of minerals that can be beneficial. So I was a bit hesitant, if you want, between a traditional carbon filter that is much cheaper and doesn't filter everything, or the reverse osmosis that filters so much that they may be filtering out too much. What do you think?
Yeah, very good point. And I've found that in my research as well, that there are pros and cons to reverse osmosis. There are, if I recall correctly, there are ways to remineralize water safely after the filtration process. But again, that's going to be another cost and another phase, you know, so something to note. In terms of microplastics, you know, we are really at the very beginning of understanding microplastics. In fact, I'm not sure exactly if there's any microplastic standard water monitoring yet, or if it can be submitted to like a private lab for testing. I think the general recommendations are that reverse osmosis is the way to go. Remember, there are plastic parts in reverse osmosis systems, so you're never going to completely get away from everything. But I will tell you, just from like I said, a consumer standpoint, reverse osmosis is usually indicated to be the best in terms of filtering out contaminants. So I think that's the right move there. And I did see a study that showed that drinking of bottled water is not recommended for long periods of time if you can avoid it. So.
Absolutely. Thank you very much. Harley is asking us if you are aware of copper sulfate impacting the development of Parkinson's.
So I have not heard about any significant Parkinson's epidemiological and experimental copper sulfate studies that have shown a consistent link to Parkinson's disease. I know that there are some experimental studies that suggest that some copper sulfate could be linked to specific types of neurotoxicity, but the data are not as strong in terms of epidemiological studies. So I think the answer for that one is it appears to be inconclusive right now, and it does not appear that there's at least epidemiologically, it has not come out to be a major risk factor.
Thank you very much. Someone is asking about COVID and the impact of COVID on PD, as well as the vaccines that were basically prescribed to help us go through the COVID phase.
So there's a lot of ongoing research right now to understand the impact of the COVID-19 virus on Parkinson's risk. I believe that research is going on right now. And what we do know is that people who suffered from really significant impacts of COVID, particularly individuals who developed long COVID, are at risk for certain neurologic dysfunction. And I think we will see as that data emerge if there's a chronic risk for Parkinson's from that. But it's a bit early to tell, unfortunately, for a disease that sometimes manifests years after the infection. But yeah, I think we're all waiting to hear on that.
Now, vaccines have been very controversial in many aspects. Have they come across as a potential impacting element in the progression of Parkinson's, or has it not been?
I have not heard of anything relating vaccine to Parkinson's disease.
Thank you very much. Yes, actually, someone is saying anonymously, thank you to you for finding understandable consumer resources. So thank you very much. Lucille Leader, actually someone I know really well because she's a famous nutritionist in the UK specialized in Parkinson's. And it is true, Lucille, that Mark and I met 5 years ago. So hello, Lucille, good to have you on the line. She's asking whether there was a research about radiation as an important aspect.
I have not heard any, it's not something that comes up very often in Parkinson's. So it's a really good question because there's different types of radiation, right? Including things like, radiation that you might get particularly from medical procedures. There's no link that I'm aware of with radiation. Obviously cancer is one that we worry about there. And yeah, I have not heard of anything. And I think that's good, right, for most of us. But if I I may look a little bit further and I can maybe follow up and send you some resources if there's anything out there
That's
that
very kind.
pops up. Yeah.
Thank you very much, Briana. Bob is making a good point. Actually, it's a question that is a leading question. He basically says that, of course, he presumes that you are using the motor nerve-based symptoms of PD to detect, to identify who has PD. But of course, alpha-synuclein can be misfolded before that, and we could have PD. Well, most of us have had PD for many years before we were officially diagnosed. So does it mean that actually the total PD population could be multiple times larger than generally reported?
Yeah, this is a great question. So I simplified, I highly simplified the work that we're doing in my presentation today, but we're very interested in trying to understand alpha-synuclein misfolding as a very early effect of environmental exposures. And you're absolutely right that understanding what is, pre-Parkinsonian or prodromal could be a much stronger population size than diagnosed Parkinson's disease. We don't have a great way yet of doing widespread testing for alpha-synuclein seeding, and so I don't know that we can answer that statistically in terms of if there are a lot of people out there that are just not sort of phenoconverting to Parkinson's because they are just not at that threshold yet. I would really like to find out. Our experimental models suggest that we do get very early accumulation of alpha-synuclein. So yeah, very good question, and something that the research community is looking at right now.
Thank you very much. Mark was saying that he had started using his wood-burning stove before we started this webinar. So someone is anonymously asking, is it advised not to use the wood-burning stove? Please say no.
I think that's again going to be something that's highly specific. It's going to depend on ventilation. It's going to depend on how efficient that stove is and whether it's working correctly. One thing I didn't talk about was wildfire smoke, which is really important in terms of environmental pollution. And so there are definitely monitors as well as air purifiers that are specific for making sure they clear out things like wildfire smoke and wood burning emissions. But yeah, definitely make sure that there's good ventilation.
Thank you very much. It depends, I guess, as well from the wood you're using as well.
Yes.
So Simon is asking a very interesting question, actually. He's asking about metal tooth fillings, which he says in the UK, and being European myself, in Europe as well, were often used with mercury. In the past, actually, he was advised to have them removed, but that has risks too. And I heard that actually, if you have them removed badly, it can basically contaminate you even further than not touching them. So first of all, should we consider removing that because of mercury? That would be a question. The other question is more for a dentist, I guess.
Yeah, I'm not a dentist, so I don't know. I don't think that there's a clear answer on this one. That's definitely something that I would talk to your care team on a case-by-case basis for. To my knowledge, metal tooth fillings have not been associated with any specific neurologic disease risks. But yeah, I can understand why you would worry about something like that, having that in your body and yeah, if you remove them and it releases a lot of metals, that could be a risk too. But yeah, certainly something to look into.
Thank you. Suzanne is asking if there is equivalent research to yours being done in Europe. Now, we had the pleasure of talking to Ray Dorsey not that long ago about, of course, the environment as well. We know a lot of the scientists in Europe. A lot of them have spoken to us, but is there anyone leading the charge in Europe for research similar to yours in the US?
Yeah, well, there's definitely great research being done all over the world with this. So I think where we have some room to grow is in the specific mechanisms of toxicity as well as epidemiology. But certainly there are researchers in Europe and the UK and actually great researchers all over the world in China and in India who are looking at environmental exposures. Where I think we could do a better job integrating is having an entity that really looks at different environmental exposures in different populations and how that manifests as disease. And so we need some kind of central, it'd be really great to have some kind of central area that we could or mechanism for us to look at how this really affects disease globally.
Thank you very much. Robert is asking a very basic question here, but very interesting one. Do you have evidence that the epidemiological links you describe, like TCE and rotenone, are causative for PD rather than just being associative factors? I'm sure you've looked at that, but actually, if you can elaborate a tiny bit.
So that is that's a very difficult question because we don't really do causative studies in epidemiology because they are usually done as retrospective cohorts. So people already have Parkinson's when the studies are conducted. And so it's very difficult to find a causal link epidemiologically. And so we talk about environmental factors as in terms of risk. And it's very it's very difficult to find a direct cause for anything, and so it's just part of the way that we have to research the disease has to be in terms of risk and not necessarily cause. And so that's even with some genetic risks, we can't always say that it caused the disease, right? And so it's something that we use in the terminology that we can't really get away from because of the way that the disease occurs, right? Idiopathic insinuates that there are many different things that could cause that.
Thank you very much. Kate has noticed that in an early slide, you mentioned that college teachers were an at-risk cluster in the 80s. I haven't seen that personally, but do you know why this occurred? Why did it happen?
Let me think back. So some of the clusters, there's a paper that Ray Dorsey published that talks about some of these clusters. And usually these clusters were due to either a source that was being emitted like around the buildings, so like a VOC or a contaminant that was leaching in, or something people were directly working with, like factory workers. In some cases, it was a dietary source, but yeah, it's usually a chemical exposure that was happening at that site.
Thank you very much. Sue is asking if there is an approximate time between exposure to TCE and onset of PD. Have you looked at basically how long does it take? I'm sure it's quite complex because it depends the quantity, the severity, the frequency, and everything. But has there been some studies along those lines?
Well, the studies have not been conducted to look at the direct relationship between time of onset. So we don't know that for sure, but it's a really important thing to be thinking about because we do believe that time is a contributing factor, right? So the exposure happens and then it takes time to develop the pathology. We just don't know what that time is, and it's going to depend on your intrinsic factors as well as the dose and the amount of TCE you were exposed to.
Thank you very much. Javier is asking about mold and fungus toxicity. Is that something that I think you've mentioned it somewhere in your presentation?
So I will say there's really not a lot of good information about there on different molds and neurotoxicity. There's some molds are considered neurotoxic and some are not. There's not a clear link epidemiologically per se, but there are some experimental studies showing certain types of mold could potentially be toxic to different cells in the brain. So I think it's sort of its own faction of research, and it's really interesting. But mold is so one of the things about like mold and PM 2.5 that I always try to think about is that they can have other health effects other than neurotoxicity, right? So we have a reason for wanting to avoid exposures that maybe are maybe we don't know that they're directly related to Parkinson's disease, but certainly from an overall health perspective, we want to try to avoid their exposures.
There are plenty of other reasons to avoid mold in your house.
Right.
Yes. So Kate is asking a question actually that is interesting. She's asking whether people who worked in the print industry prior to the digital printing and digital systems you have now were an above average risk group for PD. This is interesting because my father was in this business before and he had Parkinson's. But of course, there are many sources that could create that.
Yes, I think there have been a couple of studies that have looked at occupation and Parkinson's. But if I recall, it was very hard to link occupation with Parkinson's risk for the same reasons that we've been talking about to this point, which is that it's very difficult to find those really clear connections between a specific exposure and risk. So occupation is a really tricky one. But I know that there have been cluster cases in different industrial applications, and those papers are out there for occupational exposures and Parkinson's. So I would encourage to look into those actual primary literature studies on that.
I remember some interesting comments I got following Redorse presentations about people who were working in the army and were using basically solvents to clean up tanks, brakes, or stuff like that, and that was identified as one of the chemicals. So there is definitely a link between jobs we do and the health impact.
Right.
Someone is asking, what is the best approach to test drinking water at home? I mean, I've seen some reports in magazines, in health magazines or information magazines, but is there any way we can easily test our drinking water without having to go to a lab privately?
Well, the answer is not really. It's pretty difficult to pick up all the important contaminants with you can't really do that at home. You can send your water out for for commercial testing. There are labs that will test your water for you if you don't trust what's being told to you by a municipal source, or if you don't have a municipal source. Some people on well water get their water tested regularly. So there are places out there to do that, but unfortunately, I don't know that there's anything at home you can do that's going to give you the level of detail that will tell you all of the contaminants.
Thank you very much. Leah has a great question there. She sees many plants behind you, and she basically is asking, are plants great to have inside to improve air quality?
So the general consensus is that plants do not really clean the air, and that it's not going to be a great way to protect yourself from certain air pollutants. I have an air purifier running in the corner of my office instead. But I will say they have other health benefits, like making you feel great, and so when you're stuck in your office all day, not having to look at four boring walls. So while they don't appear to purify air per se, I think that as long as they're not toxic to your pets, they're great to have around.
They might be good at generating dopamine, which is basically also something we want to.
There you go.
We just don't want to avoid things. We want to generate good things.
Yes, that's exactly right.
The last question is a tiny bit slightly off topic, I would say, slightly only, because Nick is asking whether you have seen any link between excessive use of mobile phones and the impact it might have on the brain in general, or Parkinson's more specifically.
I've never seen anything occur, or any study occur with that. I haven't seen any data on it. If those studies are out there, I'm unaware of them. But I know that at one point there were concerns over mobile phone usage and certain radio waves that were emitted. But no, I haven't heard of anything.
Briana, thank you very much. This is all the questions have been answered. A good 20 questions answered. Thank you so much for the presentation. That was really brilliant. I haven't mentioned it to you, but some people use the Q&A to basically say how they enjoyed the presentation, how it was a great presentation. There were at least 3 people here among the questions that were thanking you for the presentation. I'm part of them too. Thank you so much. And I wish you all the best with your research. And I hope to see you perhaps next year in Phoenix.
Sounds great. Thank you for having me. It was wonderful to talk with all of you.
Pleasure. Thank you, Briana. Bye-bye. Bye-bye, everyone.